Before we proceed with the talk, I would like to invite Dr. Chong-Chin Hio, our chairperson for this international webinar series, to introduce the speaker. So, Dr. Hio, please, the floor is yours.
Yes, thank you, Tanya, and good morning all, and good evening to Dr. Tomberlin. I would like to introduce my former PhD advisor, Dr. Jeff Tomblin. For your information, I did my PhD with Dr. Tomberlin in Flies Lab from 2012 to 2016. During this period, I have learned a lot and transformed me tremendously during this time. Doing a PhD doesn't mean just another degree, but it is a life memory, experience, and friendship that lasts forever. I still remember that the road trip to attend NAFIA 2013 in Dayton, Ohio, where Dr. Tomblin brought us to Dayton from Texas. And it took us 3 days to arrive. And in between, I think we crossed 10 states. And I still remember there was a nice restaurant in Tennessee, which playing a great jazz music. And with that said, I'm extremely honored today to introduce my PhD advisor to all of you. Dr. Jeff Tomblin is a professor, Agri-Life Research Fellow, and Presidential Impact Fellow in the Department of Entomology at Texas A&M University. He is also the director of the Forensic and Investigative Science program at Texas A&M University and the principal investigator of the Forensic Laboratory for Investigative Entomological Sciences and in acronym bracket FLIES facility at Texas A&M. Research in the FLIES facility examines species interaction on ephemeral resources such as vertebrate carrions, decomposing plant material, and animal waste in order to better understand the mechanism regulating arthropod behavior as related to arrival, colonization, and succession patterns. A major focus of his research is on forensic entomology as well as waste management in confined animal facilities, as well as concern with food waste being placed in landfills. His research effort for the past 27 years have been developing methods for the production of alternate protein sources for use as livestock, poultry, and aquaculture feed from these resources. Predominantly, this effort has been accomplished through his research with the black soldier fly. Since Dr. Tomberlin arriving at Texas A&M in 2022, he has produced 15 PhD students, including myself, and 20 master students under his supervision. To date, he has edited 8 books, published 28 book chapters, and more than 180 research articles, which have been cited more than 11,000 times. Dr. Tomblin has worked with companies as well throughout the world. This includes Malaysia, China, and Australia. And he has also given presentation, for example, TEDx, on research throughout the world. So far to date, 35 countries. Yeah. And because Dr. Tomblin's commitment to protect the environment, and he, along with a group of close friends, started EVO Conversion System. This is a company located in College Station, Texas. And the mission of the company is to protect the environment while producing proteins with the black soldier fly. And EVO has partnership throughout the world as well, and welcomes other interested in learning how to become a collaborator on this exciting path towards a sustainable agriculture. So please join me and welcome Dr. Tomberlin. The floor is yours.
Thank you, Chen. That's a very kind introduction. Can you hear me okay?
Yes. Well,
wonderful.
Perfect. Yep.
Wonderful. So Chen said some very nice things about me, and I can sit here and tell you equivalent nice things about him as a student, as a person, but more importantly, as a colleague and friend. He may have indicated about the learning experience he had. It's mutual. I learned a lot about myself. I improved as a mentor, advisor, researcher. So it's a two-way street. It works for both of us.
Yeah.
And it is something I'm extremely proud of Chen and his accomplishments. He worked extremely hard and has been very productive as a student in my lab and before and after. And If you don't know this, he's been recognized with numerous awards, including the Comstock Award, which is one of the highest awards that a student can receive in the United States from the Entomological Society of America. Chin made it easy to nominate him.
Through you, yeah. Thank you for your help.
Chin. Most of it's what you did before you arrived, but thank you. That's very kind of you. So, as I put the presentation together today, what I want to do is I'm at a point in my career, I really focus on students and postdocs. So I'm going to give a presentation, which I hope, one, gives you an idea of what my program is. It's not going to be dissecting a single question in detail. It's going to be more of a snapshot of what kind of research is done in my lab and some examples of projects that are done. The goal is to give you an idea of what drives me to be a researcher, a professor, a mentor, but also to give you some insight into the questions that my lab asks. So what I'm going to do is I'm going to share my screen. Hopefully this will work. You tell me.
Sure.
Let's see if I can find it. There we go. Can you see it?
Yes.
Wonderful. Okay, so as you indicated, the title of my presentation is Janzen, Goff, and Sheppard, Icons of Decomposition, Ecology and Applied Outcomes. Where are we today? It's important that I included these names because they're important to me. They've had a major influence either directly or even indirectly. Indirectly would be like Janzen. Goff I worked with, Sheppard I worked with. So we'll get started. So the basic outline of my talk is I'm going to start broad and I'm going to talk about basics, basic research from a broad perspective, from a landscape. Then what we're going to do is narrow that down to the microbes. So we're going to talk about carrion systems in a broad sense and what's occurring in the landscape. And then we'll narrow down to where microbes and what are they doing in that environment with the carrion and how are they playing a role. And then I'm going to talk briefly about the applied outcomes. So I'm going to talk about basic research, broad, then narrow, and then the applied side. And then I'm going to go broad again and talk basic, but talk more about behavior and how that plays a role in the decomposition process indirectly. And then I'll narrow in again and talk about microbes and how they play a role in regulating these behaviors as well. So broad, the narrow, and then we'll talk about the applied side. So that's kind of the basic pattern of the presentation. And again, as I mentioned, my main drive is to really work with you students and with postdocs and give you some idea of how my career was shaped and where it is today. So to do that, I think the first thing is you should know more about who I am. This is the state of Georgia. It's in the southern U.S. If you were to see a map of the United States, Florida would be down below this area. So I grew up in deep South Georgia. This little red area is the county I grew up in. And this is my hometown. I worked in this department store right here for 3 years. And it had a massive influence on my life because the owner of the store was very involved in politics. And so I would meet people, believe it or not, ex-presidents have come through here to meet with my boss because he was so involved. So I met presidents, I met senators, House representatives, and it was all in this little store. And it inspired me to know that the world is much bigger than Ocilla, my hometown. But one of the most influential people in my life is this young lady here. This was my grandmother. This is my dad when he's about 5 years old right here. And they were sharecroppers. They grew up very poor in South Georgia. They're all barefoot. And they were running around in this area. But my grandmother taught me the importance of nature and the environment and being protective of it and conserving it. And I didn't realize the seeds that were being planted by her so many years ago when I was just a child. That resonate in my life today, much like a supervisor planting seeds in their students through discussions and engagement and research. In fact, this is me right here when I was about 2.5, 3 years old in a very similar situation. It was at my grandmother's house. You see an old barn back here with my grandmother. Well, there's an old barn here. And this is me. I grew up on a farm, basically. But other people that influenced my life were individuals such as Peter Adler at Clemson University, who was my master's advisor. He's still an extremely good friend of mine today and very much my mentor. I talk to him occasionally. I try to talk to him every year, if not more than once, just to see how he's doing. I consider him a very dear friend. And again, we talk about these people such as Dan Janzen, who wrote these papers back in the 70s on white fruit rot. And they still speak volumes to me today in terms of research. And the same can be said with people such as Lee Goff, who I worked with for 2 years at the University of Hawaii, as well as Craig Sheppard, who was my PhD advisor. In a lot of ways, you know, a lot of good things come from it. And sometimes you make the wrong decisions, but you learn from them. And that's just as important. So I always talk about my haircut back when I was a grad student. It wasn't the smartest thing to do. It wasn't the wisest. It's kind of comical. But, you know, it's part of who you are. So that you take the good with the bad. But also, it's the individuals you work with. So this is the Ecology Evolutionary Biology group logo at Texas A&M. So my colleagues still impact my life today. I learn from them. I become a better researcher daily because of my engagements with them. And then also the lab group. So the students have a massive influence on my life every day. And that's one thing I love about academia is I am pushed to improve every day. And then most importantly, my family. This is my wife, Laura, my daughter, Celeste, and my son, Jonah. I'm just dad and Jeff to them. So that brings balance. And that's something that all of you should think about is how to find balance in your lives. Now, this is the basic model of what my research is. If you look here, the key component of my research is on interkingdom communication. The idea being that there is communication between microbes and higher life. So it's really focused on this cell-to-cell communication system and how it regulates trophic interactions between microbes and arthropods and vertebrates, and how that regulates community assembly, but also within communities, how it regulates intra and interspecific competition, growth and development, individual fitness, population dynamics. So a lot of my work falls in this area, and I'm going to give you some examples of some research in my lab that's happening now or has happened in the past few years, so you can get an idea of the kind of questions that we ask. So when we think about death, there's a lot of definitions of what death is. I mean, it's the act or fact of dying or being killed, the end of life of a person or organism. This is a standard definition that you would find in a dictionary. If you look at Encyclopedia of Philosophy, it's a little bit different. It's irreversible cessation of functioning of the brain, including the brainstem. Or there are even spiritual definitions, the separation of the immortal soul from the mortal body. No matter how you define it, it is a part of your life. It's a part of what's going to happen to you, but it's also what's happening around you every day. It may be dying plants, vertebrate remains, or even soil. There's constantly birth and death occurring all around us. And we often hear a lot of discussion about the living side of things, but we don't hear much discussion about the dead side of things. And that's what my lab focuses on. What are these dead things and how does it influence an ecosystem? Now, from these, we found many ways to apply information that we learn about death, from composting and making soil, to taking animal remains and making pet food, or even distilling material to make spirits or ethanol or alcohol. So we found ways to use death to our benefit. But sometimes, bridging the basic and applied can be challenging. So let's talk a little bit about basic research and the kind of work that we're doing in the lab. One thing that's always fascinated me, this is a picture that we published in a paper almost a decade ago, but you have 6 pigs here. The thing that's most interesting about them is they were all euthanized at the same time in the same location. But what you see is they're decomposing at different rates. And that's something that's a big question to us, is why do we see such variation in the rate of degradation of these animals in the same environment? When you look at the process, it's not just recycling of these nutrients. It's abiotic and biotic factors working together to do this process. And in this time-lapse video, you see how the pig is bloating, the insects are colonizing, they're decomposing it. So these interactions are all playing a role in the recycling of these nutrients. And if we can understand this process, we can learn how to apply it so that it benefits humanity. And that's something we stress in my lab, is not just answering the basic questions to understand how process happens, but also learning how to apply that information so that we can make the world a better place. That led to the development of a critical paper out of the lab, the same one that those images of the dead pigs were published in, an annual review of entomology article. And the reason it was important is because when we look at the stages of, or the phases of decomposition as related to entomology, most work really focused on this post-colonization interval. Once insects arrive, what happens? We're focusing more on this pre-colonization interval. Before the insects arrive, what is happening to the carcass, and how does that drive succession? And this is some of the work that Chen did for his PhD, and he has published on it since then. So what do we know about carrion? I mean, if you think about decomposition processes, or decomposition studies, we take dead animals and we usually put them in cages. Well, by doing that, we're already changing the process because we're removing a major consumer of vertebrate carrion, and that is the vertebrates themselves, the scavengers. So an important question in our lab is, well, if you integrate the vertebrates, scavengers into the process, how does that influence the invertebrate population? So this is work that was done by Sam Sawyer as part of her PhD. She's defending in the next month, and she's accepted a postdoc at the University of Florida. And she did some really cool stuff, very basic approach, but really profound in terms of data generated. So the question was that she was really interested in is, when carrion are in an environment, where do they go? You know, they're there one day, they're gone the next. And we don't really know much about that process. But, you know, carrion is something that we generate ourselves, either through roadkill. So a deer hit and it's on the side of the road. We can have predation or landfills, boneyards, all sorts of things where carrion can appear. So the real question is, you know, where have the carrion gone? But that kind of strikes me as many people may not know this, but that title, Where Have the Carrion Gone, remind me of the singer Paula Cole, who had a very similar song, just not about carrion. You have to look it up if you want to know more about that. But with her objective, she was looking at different habitats. And Chen, you probably will recognize some of these habitats. And she was putting different types of carrion out. So chicks, white rats, and brown rats. And she was putting them in open terrain and wooded terrain during the summer or winter. And she was looking at scavenging rates, like how fast are the carrion removed? And of the carrion that aren't removed, how many blowflies are produced from it? So these are the two locations. One was at the Coulter Airfield in Bryan, Texas. Here's the open area, and here's the closed forested area. And the other was the Tamu Farm, which is near Snook, Texas. This is where Chen did his research. In fact, Chen, I think you did your research down here in the bottom part. But she did work where she was looking, or maybe it was up in this area, Chen.
Yeah, the above, yeah. The
right
orange
here.
circle. Yes, the above, yeah.
Yeah.
Right.
So this was an open area for Sam's work. So she had forested and open areas in two different locations. And this is what it looked like. So basically, she would put these different types of carcasses in high replicate out in these fields. And then she would go back and make observations every day and look at the rate at which the carrion was removed. And if it wasn't removed after a certain amount of accumulated degree hours, equivalent to the life cycle of a blowfly from egg to dispersing third instar or post-feeding third instar, then she would bring those carcasses back and look at blowfly production. And what she found is that during the winter, it didn't matter if it was open or wooded, all of the carrion was scavenged. What that means is for blowflies during the winter on these small carrion, they would not have access to it. During the summer, between 60 and 80% of the carrion were taken from the field, which means that there was carrion left over that could be colonized by blowflies, and we see that here. Now, this wasn't part of her research, but a secondary outcome was that we found that an invasive species was impacting blowflies as well. Now, if you think about it, if you think of this first graph, 60% to 80% consumed, which means 40 to 20% were left available for blowflies. But if fire ants are coming in and scavenging these carcasses, it really puts increased pressure on blowflies to reproduce. So that's really important because now you're talking about decreased blowfly populations in the summertime because of an invasive ant. In fact, when ants were present, they reduced blowfly production by, I think it's by 8 times. So a massive impact on blowfly production. So from a functional viewpoint, if you think of blowflies as a food resource for higher trophic levels like spiders or other animals, and if you take into account that they serve as pollinators, then there are issues there. So from her work, one thing we always talk about, and Chin's very familiar with this, is where do the microbes? So I'm going to shift gears. I just want to take a different perspective. And that another thought is, well, you have this carrion and you have flies that interact with it, but there are also microbes that are associated with these carrion resources that influence when these insects come in. And the microbes associated with these insects also influence what other insects come in, so interspecific competition. So you end up with this, what I call a Gordian knot. It's a really complex ecosystem. But one thing that's never been really talked about is where do these microbes play a role? If you have microbes associated with carrion, or is that the reason that we see variation in the succession trajectories or decomposition processes of carrion, as shown in those pictures earlier? So that gets at this pre-colonization interval again, so where we're looking at the microbes. So again, I'm just trying to give you snapshots of different types of research in my lab so you can get an idea of the kind of questions that we answered. So in this, we actually worked with mice that were xenic and axenic, so with and without microbes. So when I say axenic, there were no microbes, they're sterile. And this work was done by Casey Flint, and she's defending her PhD this fall as well. So she worked with mice that had microbes and mice that had no microbes, and she looked at fly response to these carcasses. So we use a cube olfactometer where we could release flies into the cage, and we can have treatments on either side. So we could have axenic on one side and xenic on the other. But the response of the flies is purely olfactory. So they're responding to the odors of these carcasses. And this is the device. So the flies would be on the inside. The treatments are on each side. So you see a mouse right here. The flies can go in, but they can't get to the mouse, and they get caught on sticky traps in here. And this is Casey doing some work in the lab. So she's making sure everything's going well. And then she takes these flies and she's looking at them from a phenotypic response. So she's looking at males, she's looking at gravid females and non-gravid females. So she's looking to see if phenotype drives the response. And what she found is it does. And most importantly is that the mice with microbes had greater attraction, which makes sense. The microbes are playing a major role in regulating arthropod attraction and colonization of carrion. Now, this isn't part of this presentation, but what's fascinating is that I have another PhD student, Jenny Rhinesmith, who is determining the volatile shifts on these carrion sources with and without different insects. And what she's found is that one particular insect that is competing with blowflies will change the volatile emissions so that the flies think the carrion is already degraded and they won't colonize it. So there's some massive amount of manipulation of the odors that are coming off these sources. But it's one way to avoid colonization or competition. Make your competitor think the resource is no longer available. But in these three graphs, again, is percent response on the vertical axis and days after death on the horizontal. And you see that over time for males, for gravid females and non-gravid, is that consistently the response of the insect is greater to the carcasses with microbes. Now, she went on to do another study where she looked at the choice in terms of where they lay their eggs. So she gave the flies the opportunity to colonize, and this is Cochlea macellaria, the secondary screwworm, which is a common fly in our area. And you can see in this case, this carcass has a lot of eggs on it. Well, what she found is that 90% of the eggs were laid on the carcasses with microbes. So this is a secondary behavioral response. It's not just attraction, it's also colonization. So what this is getting at, this is some of the first definitive evidence saying if you eliminate the microbes, the response to the flies completely change. The microbes associated with the carrion source are a primary driver of fly attraction and colonization. So what are the applications? We're trying to figure out this pre-colonization interval. Why is that important? Well, historically, obviously, entomology used to be used to say postmortem interval. It can be used to determine postmortem interval. Now we say it's used to determine time of colonization. But if we get a better handle or understanding of the pre-colonization interval, in conjunction with that post-colonization interval, we could potentially get better at estimating the PMI itself. So that's where all this work comes together. Now, I've assisted on over 180 cases from around the world in forensics, and they revolve around veterinary forensics, so abuse or neglect of pets, stored products where insects have come in and colonized, flies have colonized, traditional crime scene investigation cases of homicide, neglect, abuse, funeral home cases where remains are colonized, and that's not good, obviously, if you're dealing with a funeral. Even the military and federal government. So I've done work with the FBI, NCIS, and other agencies. So people that come to my lab get opportunities to get trained in forensics. And to date, about 80% of the people that are board certified in the U.S. have spent time in my lab getting training. And that's something I take a lot of pride in because I love the fact that people can come to my lab and get the experience they need that can allow them then to pursue certification. And at the same time, as I said initially, these opportunities give me a chance to learn and grow as well. So from all that work, what that led to is the creation of Iucrc. This is a National Science Foundation Center. And I was able to work with people at Boston University and Florida International University and University of South Alabama to create this center to bridge research and application. And I was able to serve as an affiliate site director for a number of years. And this has since been turned over to my co-PI, Dr. Aaron Tyrone, who now serves as the affiliate site director for the center. Now we're going to back up and we're going to talk about decomposition ecology again, but two very different projects. One thing to recognize about insects that colonize carrion is within that realm, they fall into different categories, but two I like to talk about with flies are specialists and generalists. And blowflies typically are specialists because they colonize vertebrate remains. But Hermetia lucens, black soldier fly, is a generalist. It can colonize dead bodies, but it can also colonize decomposing brown matter or plant material. And there are trade-offs for both of these different groups. They can be more successful given certain circumstances than others, but there are also drawbacks to that. And for the second part on decomposition, we're going to focus on the black soldier fly. So here's a picture of Hermetia lucens. And I've been studying the species for over 23 years now. One thing about the black soldier fly that's interesting is they have this lecking behavior, so they aggregate. And we don't know much about how that lecking behavior occurs, but we do know that there's size variation in adults. And we wanted to know, is there an impact of size on mating success of this fly that's forensically relevant and important in the decomposition process? So we know the advantages. If you're a big fly, you can have more eggs. If you're a male and you're bigger, you are more competitive typically for securing a mate. But there are disadvantages as well. It can take longer for you to develop. It takes a lot more to maintain that body size. And that's really important. So we wanted to know, well, how does adult size variation impact mating success of the black soldier fly? And this is work that a former student, Brittany Jones, published this past year. And secondarily, if their size impacts mating, how does it impact fertile egg production? Because in the end, the production of offspring is really important. Well, what we know is if you change density and you change feed rate, you generate different sizes. So this is a paper that Brittany published in 2020, but look at the adult weight on the vertical axis. When you provide them a lot of food and reduce competition, the adults are quite large compared to those that have less food and have higher competition. In fact, they can be half the size. So if you have variation in these sizes, does it impact their ability to mate and lay eggs? So we did some work where we actually put different size adults in a cage. We had small males and small females with large males and large females. And we looked at their ability to secure a mate. And what I want you to focus on is this red box. This is number of observed matings on the vertical axis, and it's broken into categories. And what you see is that large males would mate with large females, and large males would mate with small females at a high rate. And what we would also see is that small males would mate with small females, but they were less likely to mate with large females. So there's probably a female-driven selection here. So there's a sexual conflict that's taking place. We also found that when you had different sized adults in these cages, you would have a fair amount of male-to-male aggression. Over here on the right are there's the controls, the large males with large females, small males with small females. And what we saw was over a select period of time, there was about 90 matings. And there didn't appear to be a difference in terms of the number of matings in this mixed population versus these pure colonies. So the question then goes, I mean, there was a difference with the small, but that's a separate issue, and we can talk about that at another time. But if there wasn't a difference in the number of matings, you know, was there an impact? Is size really important? Well, when we look at this, for the total number of eggs, large adults produce more eggs than small adults. That's expected. And when you put them together, you get about the same amount of eggs. So there wasn't even an impact on number of eggs laid, really. Obviously, again, the small, but they're separate. We'll talk about that at another time. But here's where you see the difference. It's the impact on mating success is really on fertile egg production. Across the board, the percent hatch rate was pretty consistent, 70%. But look at this, the variation. Here is where you see the impact of variation in adult size on mating success. And that is, the variation was like 8 to 10 times greater in the mixed population, meaning that the hatch rate was highly variable. This is where you see the impact on mating success. This is where you see the outcomes of having variability in adult size. So what we learned is that large males, in the study, we learned that large males have an advantage over small males. And we learned that egg deposition and hatch was highly variable for the heterogeneous population, which is, when we get to the applied side, really important. But why such variability? Well, egg capacity, right? So if you have a large male mating with a small female, you may get high fertilization rate and high hatch rate, but it's fewer eggs. Versus if you have a small male mating with a large female, they may only provide a small amount of sperm, so only a few number of the eggs hatch. And we know that these adults typically only mate once. We also know that the adult age could be a factor. So as they get older, their sperm amount decreases. And we know that malnutrition of the small adults could also potentially be a factor because it could reduce the quality of the sperm that the males produce, but also the quality of the eggs produced by the female. And again, on the sperm load, it's really important. Alini Malloway, former PhD student, just published this paper. We found that the sperm number drop off drastically in these flies. And if they're small, they already have a small number of sperm. And if they're dropping off quickly, that means by the time they mate, they may not even have sperm present. So that's one of the basic areas that we're studying is what drives mating in oviposition. So here's a female laying eggs. But that led to some other questions of, well, once females have laid eggs, you know, where do microbes play a role? And I'm going to go over a few data to talk about where microbes are important in this process. So the first thing we did is we had to go back and we had to look at the microbial communities associated with the different stages of development, from larva to pupa to adult to egg. And we did that at different taxonomic resolution scales. Chin's very familiar with this. And we found that there's a lot of variability, and that can be challenging in terms of application of microbes in such studies. We also found that, like from some previous work, is that a lot of these microbes are really important in terms of the development of the insect. And we worked with a few of the bacillus bacteria that were isolated from the black soldier fly, and we found we could actually use them as a probiotic to enhance development. So there were certain strains that actually had larvae grow bigger, like 40% bigger than the control. In other instances, they suppressed growth. So, I shouldn't say suppressed growth, they accelerated development. So they could increase the weight and they could decrease the amount of time to gain that weight. So these microbes are really important in the nutrition of the insect. We also found that many of these microbes could be used to enhance oviposition. So we took microbes from the eggs and we cultured them and placed them in this cardboard, and you can see which cardboard had the microbes. All of the females are laying eggs there versus the control. So we're looking at where the microbes play a role in the black soldier fly biology. So why is that important for sustainable ag? Well, one thing to recognize about the black soldier fly is it is the only insect that's approved globally for use as a feed ingredient, because it can reduce waste, produce protein, and then that protein can be used as a feed ingredient. So where do the data that I mentioned on behavior, especially with regards to mating, play a role? Well, it's because the adult is housed in this cage in these artificial environments where they're forced to mate, or they're forced in these small enclosures where they do mate. Hopefully this video works, but this is what it looks like. Can you see this? So there's a light that regulates the mating. So watch when they cut it back on.
Well, I think from my side, I can't see the video. Yeah.
You can't see the video?
I cannot. Yeah, cannot.
Okay.
Right.
But that's the mating behavior. So these artificial lights drive them into these mating swarms, and there's a lot of selection taking place. In fact, right here, you can see a pair that are engaged in the second stage of this mating behavior. So the male is trying to mate with the female. Now, this is another video, and hopefully it comes through, but it's in slow motion. So the light is on, the adults are swarming, and decisions are being made on mating. When the light is on,
Excuse me, but we can't see the video.
So we don't know much about this mating behavior at all. And I want to thank Noah Lemke, the new PhD student in the lab, for providing these videos. But we're trying to figure out where the size factor plays a role. We know it does. We demonstrated that with Brittany's work, and now Noah's diving in to understand this process a little better. So again, why is this important? Well, if we look at what's happening around the world, we live in a paradox of we're expecting to have 10 billion people on the planet. We need to produce more food so we can feed them. But believe it or not, 40% of the food we produce, we waste. In fact, if you look at fruits and vegetables, 52% of what we produce is wasted. And for seafood, 50% of what we produce is wasted. So we have this large amount of waste that's being produced. Now, one thing to recognize from food waste is, regardless if you're talking about beef, pork, chicken, or you're talking about fruits and vegetables, 1.5% of all the greenhouse gases produced are from this food waste. Well, the black soldier fly can be used to recycle this waste. In fact, it can recycle that waste to produce these larvae, which can be used to produce fish meal or a replacement for fish meal, and used in aquaculture. But it can also be used for pet food, swine, poultry, and various other livestock. And what we're seeing is that based on work that we did in the past with people from Italy and people from Canada, researchers, colleagues, that they can digest this waste and they can keep it from going into landfills. We can also use it to reduce other sorts of waste, such as livestock waste. So Chelsea Miranda finished her PhD not long ago. She looked at this, and what we found is that they can do a massive amount of bioconversion of dairy, swine, and poultry waste to produce biomass. This has also been done by other people, such as Larry Newton at North Carolina State. The point being is black soldier fly can digest this waste and produce products of value. And that led us to develop another National Science Foundation Center that just got funded this year. And we're working with companies from all over the world, including from your part of the world. And the goal is to bridge these industry with academics like ourselves to conduct research. And I just really want to recognize Mississippi State and IUPUI as co-leaders in the development of the center. So in both instances, in forensics and in sustainable ag, we've been able to do basic research and build it for applications. And that's a big part of what my lab is. You're a basic researcher with applied outcomes. And I really stress that in my lab. And this is just a list of the different companies that are working with us. And one thing I like about this center is that we work with companies that are startups, but we also work with large companies like Tyson Food or Mars Corporation. So this is really a lot of fun because we're working with various size companies. So when you think about my lab, the Forensic Laboratory for Investigative and Entomological Sciences, we do a lot of different things, but it all falls under the umbrella of decomposition ecology. And what I would stress to all of you students and the postdocs that are in the audience tonight is don't limit yourself, don't shy away from a challenge, and be fearless. Chase your dreams, find balance in life, and pursue what you enjoy as a career perspective. If you do that, if you stay focused, you can have a very successful career. So at this time, I'll be glad to take any questions.
Thank you, Dr. Tomberlin, for your impressive presentation. Now we will proceed with the Q&A session. I will read the question that the participant will post in the chat room. And also for those who want to ask some question to Dr. Tomberlin directly, please do so by turning your mic on. We always welcome an interactive session.
Yeah, thank you very much, Dr. Tomblin, for your always awesome and inspiring presentation. I really miss your voice and your presentation. Yeah.
Thank you, Chen. Chen, could you see the videos?
Well, sorry, both videos I couldn't see. Yeah,
I'm sorry.
unfortunately.
I'll be glad to share
right.
those videos with your lab if you would like to see them. But it's basically in the cage and showing how the flies are engaging one another.
Wow. All right. Yeah, that's cool. I would love to see. Yeah. Thanks again. Right. So maybe I can begin with a question. I was really interested with your research that putting mice with microbes and the other mice without microbes. I just have one simple question. Did you sterilize the mice with autoclave? And how do we know that the microbes are not present in the internal organs? Yeah, that is my simple question.
That's a great question. But Chen, thumbs up, you could see all of the slides? So all the images, though, you could see?
Yes, okay. Yeah.
So that's a great question. So what we do is we work with a medical school. They actually have a colony of mice that have no microbes, 0 microbes. On the inside, outside, it's completely sterile. So they euthanize these mice over time because they don't need them. And they'll sell them to us. And they put them in sterile containers, and they're shipped to us sterile. And we also tested them over the course of the experiment to make sure they remain sterile. So we take them in a biosafety cabinet and sample them. So the beauty of the working with the medical school is, in the future, we can actually tailor the mice to have certain microbes. You can inoculate them, so you know exactly what's in them. In fact, you can actually tailor these mice to specific diets. Like you could say a Mediterranean diet, you could say an Asian diet, an American diet. You could tailor the mouse to have the physiology response of the diet to the mice, and then how they engage the microbe, and then examining how that impacts decomposition.
That is really cool. That can answer a lot of questions. Yeah, because I always believe that the changes in microbiome either outside or inside, may affect the behavior or the pattern of succession. I always
Yes,
believe
I
that.
agree.
Yeah.
I agree.
That's
And one thing I would encourage you to do is
Yep.
talk to different medical schools in your nation. Someone may have one of these colonies, and when they euthanize those mice, they have nothing to do with them because they want to work with them when they're alive. So you may be able to get access to such resources.
Yeah, that's great. I try to look around. Yeah, what may I know how do they euthanize the mice? Is it by CO2 chamber or they by cervical dislocation?
It's dislocation.
Dislocation. All right. All right. That's all my question. Thank you.
Thank you, Dr. Hill. Thank you, Dr. Tomberlin. I have a question about also the cage that you use in this experiment. Why is covered all by black clothes, or I don't know what is this, but I'm wondering about the reason about that.
Right. So we use the black cloth to make sure the flies aren't distracted by anything outside the box. So they can't see anything. All they can do is we have LED lights on top, so no heat. They don't produce any heat. And the flies don't see anything. So all they're doing is they're responding to odors. And you can build, if you want to do behavior work, these things are very easy to build. They're out of plexiglass, and you can just wash them in between treatments. And it's great ways to do all sorts of behavior research. You can do species interaction because we did work where we did things like I had a former student, Micah Flores, and he was dealing with predator-prey interactions. So Chrysomya rufifacies, and looking at how it impacted the native species response. And his results were fascinating because he showed that if a blowfly, a prey blowfly, had to make a choice between colonizing a carcass with its own larvae in late stage of development or a carcass with a predator in late stage of development, it was more attracted to the carcass with the predator. And we think the reason being is twofold. One is that in terms of competition, intraspecific competition would be more devastating because what you're competing with is eating what you need. So if you colonize a carcass that has late developing predators, well, they don't eat everything and they may leave something for you. Secondly, they're not interested in you because Chrysomya rufifacies isn't going to eat eggs or first instar prey. So we did not expect that, totally outside of what we expected. But that cube design, you can do all sorts of behavior work. It's fun. Generate a ton of data, look at species interactions. If you're not doing it, it's an easy way to do experiments in the lab.
Thank you so much, because I was interested in doing something about selection for the different type of bites. So it was nice to see your design of the cage.
Sure.
Thank
we
you.
have a paper. We have a paper that was published on that design. Just came out last year. And
I think I read it. I have this paper. I'm not sure if it's with Hermetia or with...
No, it was with Cochlear macellaria, I believe.
Okay, yeah.
Casey Flint did it, and it really got into how to validate your population with that design. So
okay. Thank you so much.
Mm-hmm.
Any question more for okay, some is raising their hands. Please turn on your mic. Adila?
Yeah, hi. Can I go first?
Yeah, sure.
Hi, Dr. Tomberlin.
Hey, Adela, how are you?
Doing good.
Good to see you.
Yes, me too. Okay, Doctor. Thank you for your presentation. Actually, I wanted to know, so where are we today? Because we have advancement in the research, but as we know, from the replicate, control, disruptive environment, it is very different compared with the casework. So how can we relate between this research, this ongoing research with the casework?
Yeah, so I think what's really key is validation work. So if you do research and you can do validation work, you should. And Chin was part of projects on this. We did this at A&M, where we validated results. So we did something on Hermetia lucens. But the idea is, if you say colonization of a body occurs at this time, and you suspect the postmortem interval is so much longer, you need to do some work to validate that. And there's ways to do it, right? I mean, you can take and put carcasses out, and you know when you put them out, and you can watch them for colonization, and then you can relate your estimation of how old those larvae are on that body to when it died and when you put it out. And you can find out how precise you are. And you can do that in all sorts of environments, different times of year, different conditions. And you should do that. We should do that, because the more validation work we have, then eventually we can start doing meta-analysis, where we take different data sets and bring them together. And then you can find out for a region, what is the variability and what is the accuracy and precision of your estimates. But it starts with one study. You know, you take and put carcasses out in a forested area, say a palm forest, and you look at when things are colonized, and you make estimates based on data sets, published data sets on the species of interest. Then you do those same studies in different months of the year, different locations. Eventually, you'll accumulate enough estimates to really find out how precise you are.
All right, cool. Thank you, Doctor. Thank you.
Yeah, sure.
Hi, Dr. Tomberlin. Can I ask the next questions? Nice to meet you online.
Sure. Nice to meet you.
I have read a lot on your work, and I'm very interested in finding more about BSF mating. I'm particularly interested in the mating behavior. So I would like to know, from your experience, what is the average matings by the males? Because we know that the females always mated probably just once in their lifetime. So how about the males? What is the average matings performed by a male?
So I'm going to speculate in the wild, males typically mate once. In a colony, they will mate multiple times. 15% multiple matings. I believe males that mate a second time have zero sperm. So it's a form of mate guarding.
So in that respect also, for investigating the mating behavior in captivity, so what is the recommended minimum size of the cage from your experience that will enable a more natural setting for the mating to take place?
So the design has not been sorted out yet. We don't know.
So there's not much has been done on that area, is it? So have you actually also looked at the durations of the mating between, means not the courtship, but the whole durations one they copulated, and how long they has been actually lasted for a successful mating? Yeah.
That's a great question. I'm talking with my new PhD student about that, is to capture pairs when they mate, look at duration, and then weigh the adult to see how big the male is, how big the female is. Have they mated more than once? That sort of data. Beautiful data, but no one has collected them yet.
I guess probably it's hard because they mate in the air, right?
They will grab each other in the air, but they fall to the ground.
And fall, yeah.
Mm-hmm. And they're usually, at least for our population, very gentle. So you can pick them up and you can put them in a container and wash them.
Okay. So do you think that because we can rear them in a semi-natural environment, in a semi-natural environment, which normally we started with, but I think that recently they also find a way to rear the insect within the lab with artificial conditions,
Yes.
right?
Yes.
In terms of the lighting, I guess. Probably, I think to study mating, it will be much better to do it under the semi natural condition rather than in the very much artificial with the lighting and all that, right?
Yes. If you look at this screen here, let's see. So you see the screen here?
Yeah, yeah.
The cage? That's the artificial cage with an artificial light on top.
Mm-hmm.
So those artificial lights allow you to do really controlled work. And the cages, like the one in that image, are typically what you would see in a factory.
So that artificial lighting probably more suited for rearing, for the purpose of rearing it, probably not for research purposes. I mean, in terms of mating.
It's very good for it because you turn the light on, they swarm and mate. Turn the light off, they stop.
Okay, so my last question would be I would like to see the video that you're trying to show us just now.
Sure.
I was waiting, but couldn't. But it's regarding the mating as well, right?
Yes.
So it would be nice.
It's a video of the swarm.
Yeah.
Where the mating occurs, and I can talk to I'll talk to Chin about sharing the video, and then he can make it available.
Okay, all right. If it is not, if it is available in the internet or YouTube, then we can actually visit it as well.
Yeah.
Okay, nice talking to you. Thank you.
Pleasure meeting you.
Right.
Thank you so much. We have more hands raised. Asmira, please. Do you want to ask us something?
Yes, Tomlin, very interesting presentation. But I'm interested in the rearing of the black soldier fly. So when rearing black soldier fly, have you ever had fungi problem, especially when trying to hatch the eggs? Because in my case, I placed the larvae and the eggs in the same incubator, but it seems like the larvae are not affected by fungi, but the eggs they did not hatch.
So the eggs, the female will lay infertile eggs. That's the problem, is sometimes they're fertilized, sometimes they're not. And that's why we did the adult size experiments. And the most important thing with the eggs is they typically hatch in 3 or 4 days, but you have to maintain them at high humidity and a warm temp. So 27 degrees Celsius at minimum, and relative humidity at 60-70%. If the eggs are fertile, they should hatch. But if they're not, you may end up with a fungal growth on them.
Thank you.
Yeah.
Thank you so much. Sarabjit?
Yes. Hi, Dr. Tomberlin. Thank you for the great presentation. And, my question is on black soldier fly, because it's a black soldier fly is really new for me, especially for me. So I just want to ask that why we use that some like a rotten food or manure or wastage food for the establishment for the black soldier fly colony establishment.
Why do we use it?
Yes.
Well, you wouldn't use it for the colony. You just use it to digest. So to produce the biomass of the insect for feed. For your colony, you would go with something more conservative, more consistent, like a standard diet, because you want to protect your colony. I always say the colony is the heartbeat of the company. if the colony dies, the company stops, right? So the colony, you would feed a standardized diet. The food waste, just for growing larvae. Because you may get bad food waste and it could kill the larvae, and you don't want to do that to your colony.
Okay. Okay, thank you. So my second question is on that. I saw that you, in your presentation there is a black a black box that I think for the behavior of the flies. That when I was in Dr. Helen LeBlanc's lab, they conducted she conducted an experiment of Y tube experiment. So is it same with that? Both are similar, or there is a difference?
That's a great question. So there are differences. Our device is passive, so air accumulates and spreads into an area. A Y tube typically has a motor that will pull the air. Also, in our cage, in our cube, we can put hundreds of adults, where in a Y tube you may use one adult. And there are pros and cons to that in terms of the statistics, the interpretation of the data, and you have to decide what you want to do. We like the cube because we can generate a lot of data real fast. The Y tube is really challenging. It's really strong data, but it's really challenging. And I've had students publish with Y tube. Adrian Brundage, a former PhD student, used Y tube, and I felt so bad for her because it just takes so much effort.
Mm-hmm. Yeah. Okay. Okay. Thank you. Thank you.
Yes.
Thank you so much, Dr. Tomberlin, for your answers. Any more questions from the audience? Okay, if there is no, okay, we have one in the chat box. May I know what makes you so interested in studying insects?
Yeah, so there's a lot of, I think, reasons behind that. I think growing up in nature, I grew up in the country on a farm, so I was around it a lot. And I think it was part of who I was before I even knew it. And then when I was at a university, I took a class on entomology and I learned about forensics. And I said, I have to do this. And I fell in love with it and I pursued it. And I had people say, you don't want to do that. Don't do that. And I ignored them and I stayed focused on what I wanted. And I chased my dreams. I never looked back. I never took no for an answer. And I just stayed focused and I pushed through. And I think all of that played a role. When I talked about my grandmother, you know, she was not an entomologist by any means, but she was a very outdoors kind of person. And I think being around people like that had a big influence on my life.
It's a great answer, Dr. Tomberlin. Yeah, I feel that. Yeah.
My grandmother was the most influential person in my life. I mean, my parents were very influential, but my grandmother, I would come home from school and go straight to her house every day, go to her house.
Yeah.
Thank you so much, Dr. Tomberlin, for your sharing. I think all the people that work with insects, we have similar stories that
Yes.
we can share about how we started.
Fall in love in this. Yeah.
In this field, or in forensic entomology. I would like to ask you the last question because the time now is a bit late. Actually, we know where are we today, but in your opinion, what is the big gap of knowledge that actually we have in the composition ecology?
Yeah, I think we do not have a full appreciation for the time from death to colonization. We don't know what causes that variation, and we're not good at predicting that time interval. If we ever get to the point where we're good at predicting the pre-colonization interval, I think we're very good at the post. I think we can do post-colonization interval. If we can get to that same level with the pre, I think we will elevate forensic entomology to truly be able to say postmortem interval.
Yeah.
I agree.
It's
Yeah.
true. I agree also.
Yeah. The challenging part is the pre-colonization interval. I face it during my report writing. This is very much true. Yeah.
Yeah. Okay, thank you so much, Dr. Tomberlin, for your answers. If there is no more questions, please allow me to close this seminar now due to the time constraint. And don't forget to sign the attendance form to get the e-certificate. For those who are interested to join us in the next webinar series, the date is set on 21st October by Dr. Stefano Banin from Università di Genova in Italy. I'm looking forward to meet all of you again in our next webinar. Again, I would like to thank Dr. Tomberlin for agreeing to become our guest speaker for today, and the participants for joining us, and remember to stay safe. Goodbye, everyone.
Thank you, Dr. Tomblin, for your time.
Thank you.
Yeah, we enjoy your presentation.
Thank you so much. And I just want to say this to everybody is I love the internet because it makes me feel like I'm right there with you. I look forward to COVID restrictions being lifted and one day soon being able to meet you in person.
Yes. Thank you, Dr. Tomblin.
Thank you.
Hopefully one day. Yeah.
Thank you.
Be soon.
Have a wonderful day.
Yep. Have a nice evening to you. Yeah.
Bye-bye.
Bye-bye. Thanks.
Bye.
Everyone. Thank you.
Bye.
Bye. Yeah. All right. Okay. So good job. That is the webinar by my former PhD advisor. Yeah. So hope you all enjoy it. All right. Let's looking forward to the next webinar. Right. All right, everybody. Yep. Take care and stay safe. Okay. Bye-bye. Goodbye.
Bye. Bye.